YouTube is already started.
Okay. Sure, sir.
So you can proceed.
Sure. Thank you. Good morning, one and all. Today, on the very auspicious day of 1st January 2023, I'm very happy to say that Microbiology Society of India is continuing this PhD webinar series. First of all, I would like to invite our All India President, Dr. Arvind Deshmukh sir, to speak few words.
Okay. I'm very happy today that new lecture is going to be delivered today. And Dr. Jalpa from Gujarat. Really, I'm very happy that is all this young generation is enthusiastic and ready to deliver the lectures so that maximum students in India will get the benefit. The reason is that these lectures are stored on YouTube and will be made available. Today's lecture is a really excellent lecture on nanoparticle. Topic is a nanoparticle and related with the West. Dr. Jalpa will deliver the lecture. As a president of Microbiology Society, it is my duty to wish all the best, and again, on occasion of New Year beginning, I wish Happy New Year to all microbiologists of India and also to Jay for this series and also to Dr. Jalpa. She is the first lady in this year on our platform. I congratulate her and wish all the best. Happy New Year to all. Thank you. Thank you very much.
Thank you very much, Arvind Deshmukh sir. Thank you. Today we are having our 22 speaker, Dr. Jalpa Magodia madam from Junagadh. I would like to invite Dr. Jalpa ma'am to start her lecture. Ma'am, please.
Yes, very warm good morning to all of you. Today is the very auspicious day for me and all of you, and Happy New Year to all. So we started to development of cellulose-based and green nanoparticles, silver nanoparticles embedded biodegradable plastic film derived from agricultural waste from utilization of food packaging. As we know that plastic is ubiquitous, it is used in everything from our shampoo bottle to your golf shirt. Unfortunately, traditional petroleum-based plastics are notorious contributors to landfill waste. So, according to a 2017 study in Science Advancer, of 8.3 billion metric tons of virgin plastics produced since 1950, and about 6.3 billion metric tons have been disposed of in landfills or the natural environment. If this trend continues, that number could increase to 12 billion metric tons, and this is very harmful to our Mother Earth. So the next. So we can use the natural biopolymers like cellulose for their plastic alternate of the plastic, and municipal solid landfills are assigned area of the land and exhibitions that receive household waste. Landfills are the only designed to store waste, not break them down. Unfortunately, the rate at which we are producing waste outpaces the rate of natural biodegradation. The disposable of non-biodegradable plastic is unsustainable because they often follow a linear economical model where virgin produce are manufactured, used once, and disposed of, accumulating in landfills or the natural environment. While recycling is the one and only solution to divert waste from the landfills. Next. So there are many types of cellulose-based polymer we can use for the alternative plastic like cellulose deconstruction, monomer-based polymers, natural cellulose fiber, or nanocellulose-based polymers. Next. In nature, cellulose is most commonly obtained from trees and cotton, and about 180 billion tons of cellulose is produced per year. Traditionally, the demand for cellulose has been met by wood and cotton. Therefore, necessitate a search for commercially viable alternative to plant cellulose, which is free from this problem or difficulties and can fulfill the cellulose demand and the most important biopolymer needed. Next. In my research work, I used sugarcane bagasse for the synthesis of cellulose-based plastic, and then I used synthesis of silver-based nanoparticles. And combination of these silver nanoparticles and cellulose polymer, I synthesize a new biodegradable plastic film, which is used to alternate of food packaging. Next. In past 2 decades, the use and production of plastic in the world have been tremendously increased. Plastic production has increased from 0.5 to 260 million tons per year since 1950. So we can utilize the further analysis of gelatinous mat or cellulose-based material was similar to the plant cellulose in chemicals characterization. And as a microbiologist, I used a microorganism which can also synthesize a cellulose-based polymers like acetobacterium or Acetobacter xylellum, which is used for cellulose formation in living system. Next. How can we utilize cellulose-based polymer for plastic production? There are several various types of method, from plant extraction and utilization of thin film production, filtration method, photoelectric device, biomedical engineering. These all are the very tremendous in market. Next. The term biodegradable material is used to describe those material which can be degraded by the enzyme action of living organism. Such bacteria are like all microorganisms in aerobic or anaerobic condition. They can degrade the biodegradable plastic and solve our problem. Next. You can see the landfills are totally covered by plastic waste. Next. Nowadays, efforts are being made for production of biodegradable films for more effective protection of food products. This research is aimed at converting some of these unwanted agricultural waste or byproducts to commercially useful products such as pulp and cellulose acetate. My work has the potential of providing new market and applications for low value and utilized agricultural waste by converting into cellulose acetate. Next. There are various sustainable properties of cellulose like renewability and high surface area to volume ratio, biocompatibility, improved mechanical properties, low density, cheapest cost, optical transparency, adaptable surface chemistry, biodegradability, and most important, sustainability. Next. So now, sustainability in industrial ecology or eco-efficiency and green chemistry are guiding the development of next generation of materials, products, and process. This biodegradable plastic and bio-based polymers product based on annually renewable agricultural and biomass feedstock can form the basis for a portfolio of sustainable and eco-efficient product. Next. Next. So my work is divided into total 6 plan of work. In first part, I prepared cellulose from the sugarcane bagasse by chemical and microbial method. In plan B, isolation and characterization of cellulose producing potential bacterial strain. Totally I isolate 22 bacterial strain, and from one Acetobacterium caurococcum is the potential producer of cellulose. After third stage, purification and characterization of bacterially produced cellulose was done. In fourth stage, bacterial synthesis of silver nanoparticles and its characterization was done. In next stage, synthesized silver nanoparticles are incorporated into cellulose-based polymer, and in last, study of prepared plastic in food packaging. Next. Normally, cellulose production was done by chemical and microbial method from sugarcane bagasse. In chemical method, I used dewaxing of sugarcane bagasse, after that delignification, then acetylation, then thalattalization of sugarcane bagasse. Or we can also use microbial method. In microbial method, first we can isolate cellulose producing bacteria, and then cultivating and screening of cellulose producer from agricultural waste. And after that, production of cellulose was processing and purified. Next. You can see the flow chart of cellulose extraction from the agricultural waste. First, agricultural waste was dewaxing in Soxley apparatus, and after that, delignification of sugarcane bagasse was done, and then acetylation, then thalattalization, and after that, we can get cellulose acetate phthalate, which is widely used for the preparation of biodegradable plastic film. Next. In second stage, microbial strain was isolated from agricultural waste, and then its morphological and screening test was done. Cultural characteristics also done, and lastly, I characterize microorganism based on their 16S rRNA genetic characterization, and phylogenetic tree development was also done. Next. Total 11 sample I used, like some fruits and vegetables, and from these samples I found one microorganism that is potentially produce cellulose from agricultural waste. And this isolation I use screen and screen media for their isolation. Next. For that screening purpose, I used cultural characteristics like growth on agar medium, growth in liquid medium, pigment production, morphological characteristics like gram staining, cell shape arrangement, and the genomic characterization, 16S rRNA, and I also done statistically proved hypothesis. Next. For cellulose production, strain was inoculated into hysterin and scram media, and after incubation, we can use their cellulose production. Next. All the pellicles were found on the hysterin and scram media produced by the bacterium. It was purified and used as a biodegradable plastic production. Next. For the purification and characterization of cellulose, two methods are generally used. First one is acid treatment, and second one is the alkali treatment. In acid treatment, we can use sodium hydroxide, potassium hydroxide, or any of the sodium bicarbonate. And in alkali treatment, we can use organic acid for the purification of cellulose found from the bacteria. Next. All the methods are used for the purification of the cellulose, and after that, this purified cellulose was incorporated in film production. Next. The major challenge is the production of silver nanoparticles from the bacteria, and after the synthesis of silver nanoparticles, these nanoparticles are incorporated into this cellulose-based biofilm, and this film is utilized as a food packaging. In synthesis of silver nanoparticles, I use Bacillus subtilis, and then characterization of silver nanoparticle was done by various methods like scanning electron microscopy, then EDEX, then particle size analyzer with zeta potential, then UV visible spectroscopy, then FTIR, and last one is antimicrobial activity. Next. There are two portfolio for the synthesis of silver nanoparticles, top-down method. I use this top-down method. For the synthesis of silver nanoparticles, Bacillus subtilis was used, and solution of HgNO3 and the culture as ethanol water extraction was used for the synthesis of silver nanoparticles. Next. UV visible spectroscopy was the first method I used to characterize the silver nanoparticles is properly done or not. After diluting 1 ml of a nanoparticle solution, I found 300 to 700 nanometer using UV visible spectrophotometer. Next. Particle size analyzer with zeta potential, also characterization was done by EDEX with dynamic light scattering, and the same analysis was undertaken to know the size and morphology of the silver nanoparticles synthesized using Bacillus species. Next. FTIR was also the another method for the synthesis of, characterization of silver nanoparticles to identify the associated biomolecules with silver nanoparticles. Next. Antimicrobial activity of biosynthesized silver nanoparticles. For this, I choose various organisms like E. coli, Pseudomonas, Aspergillus niger, and Aspergillus flavus for the antimicrobial activity checking. Next. Antifungal activity was also done in the presence of Aspergillus niger and Aspergillus flavus by using PDA media, potato dextrose agar, and various types of zone of inhibition was found from the synthesized silver nanoparticles. Next. Last one is incorporation and characterization of silver nanoparticles embedded biodegradable plastic. I used incorporation of silver nanoparticles to embedment of in cellulose-based film, and this film was utilized for the food packaging of various food samples. Next. For the preparation of polythene bag incorporated with Ag silver nanoparticles, I use simple casting method in which 2.5% of solution of cellulose in acetone was prepared by stirring 100 rpm for 10 to 15 minutes using a magnetic stirrer. After that, polyethylene glycol as a plasticizer was added into it, and then after bacterial synthesized silver nanoparticle was added to it. Next. After casting, the cellulose film was done for characterization for various physical and chemical properties like tensile strength. Next. Scanning electron microscopy of developed plastic film was also done and checked by scanning electron microscope. Next. In mechanical properties like tensile strength and elongation at break for this cellulose film was determined by ASTM method, and the sample was overgripped on the device with a gap of 1 mm and cooled away at a speed of 0.1 mm, load of 1 Newton. Next. Food packaging study, I utilize various types of criteria like determination of pH of the packed food, total plate count of packed food, determination of pH of the packed food, biodegradation of prepared plastic, bioefficacy of prepared plastic film, and total plate count of packed food. Next. Application of this prepared plastic is in present study. I use total 5 different food samples like apple, bread, potato, tomato, and Balaji masala wafer for their food packaging study. Next. In total plate count, packaged food are used for determination of normal flora present in their food. And in approximate TPC analysis, the calculation of the number of bacterial colonies present in the sample with dilution is needed. Next. In statistical analysis of total plate count of packed food sample, approximate 0.1 ml portion of the sample dilution was spread on duplicate sterile plates. And after incubation, we used Turkey's multiple comparison test for the statistical analysis. Next. First, determination of acidity of packed food. Generally, the acidity of the food samples means the total acidity or titrable acidity. I use titration method for their acidity count. And content of this acidity was determined by volume of food sample with standard alkali. Next. Similar way, a determination of pH of packed food was also determined by pH meter of the samples. Next. Bioefficacy of developed biodegradable film. To determine this antimicrobial activity of silver nanoparticles incorporated in cellulose-based film was tested against 2 organisms, that is E. coli and Bacillus subtilis bacteria. And for fungi, it's Aspergillus niger and Aspergillus flavus by disk diffusion method. Various types of results were found. Next. Biodegradation study of developed biodegradable plastic film was also done. Different plastic samples were placed under soil in the tray, and after regular intervals, degradation of plastic was checked by total plate count method. Next. These are the results of my work. This is the flask of Hestrin and Scram media, in which you can see that the cellulose biofilm or cellulose pellicle was upper the layer of the media. Next. Now this pellicle or pellet obtained was the process by treating this with acid method, and after the purification, this cellulose was utilized in film production. Next. Various samples are used for the cellulose production. In chemical method, I use 2 samples, sugarcane bagasse and groundnut cells. And for microbial method for cellulose production, I use various vegetables waste and fruit waste. Next. From this, isolate JR7 with sugarcane bagasse showed maximum bacterial cellulose production, that is 0.98 gram per liter, followed by banana from 0.88 gram per liter also showed best yield of cellulose. Next. After that, this isolate was characterized culturally and morphological characterization. In this, growth on agar media, growth of liquid media, Gram's reaction, morphology, and arrangement was also done. Next. These are the isolates. You can see that Gram's staining reaction and growth on Hestrin and Scram agar plate. Next. Now the identification of this JR isolate 7 for this 16S RRN analysis was done. Then next. This is the sequence alignment report of 16S RRN sequencing of isolate JR7. Next. So from this sequencing method, and sequence alignment report, I found this my isolate is identified as Azotobacter corococcum, and it is the potential producer of cellulose from the agricultural waste. Next. This one is also the report of 16S rRNA sequencing. Next. 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 Phylogenetic tree was also developed based on this evolutionary relationship of taxa, and corococcum having a 99.99% similarity with my isolate. Next. Next. This is the same image of cellulose produce was studied, and this cellulose was utilized in plastic film production. Next. This are the nanoparticles utilized for the food packaging. Next. UV visible spectroscopy of biosynthesis of silver nanoparticle was also checked at 440 nanometer. Next. This is the graph of UV visible absorption spectra of silver nanoparticles synthesized by Bacillus species. Next. This particle size analysis and zeta potential analysis show that the nanoparticles I synthesized from bacterial species have a very tremendous use. Next. This is the same image, scanning electron microscopy image of silver nanoparticles. Next. This one is the index image of biosynthesized silver nanoparticles from the Bacillus species. Next. In FTIR analysis, I found wave number of 2956.56 centimeter correspond to asymmetric stretching of this plastic film. Next. This data is the FTIR analysis of biosynthesis nanoparticles. Next. This one is the film I produced from cellulose, and by incorporating the silver nanoparticles into it. Next. I produced totally 5 types of cellulose-based film. In first one, I added 0.0058% of silver nanoparticles. In second, I added 0.0075 weight%. In third, 0.01 weight%, and in fourth, 0.0125 weight%, and in last film, a cellulose-based plastic is added 0.15 weight% silver nanoparticles. Next. Now this film was utilized for the active food packaging, and total 5 samples were packed within this film. And I found that the normal plastic bag, which is used in plastic packaging, and from my prepared bag, there is a very long shelf life of this food samples. Next. Next. In statistical analysis of total plate count of packed food, an ANOVA test was conducted to compare the total plate count in normal polythene bag and cellulose bag with different concentration of silver nanoparticles was also checked. Next. These are the statistical result. Next. To check bioefficacy of the developed plastic film, I utilize E. coli, S. aureus, Bacillus subtilis, Aspergillus niger, flavus, and from these, various types of zone of inhibition like 2, 3 mm, 2.1 mm, 2 mm, various types of zone of inhibition respectively was found. Next. Now, the main goal of this is how can we biodegrade this polythene bag? So this film, developed cellulose-based film, was degraded in normal condition into soil within 72 hours. Next. To check the silver nanoparticles hazardous effect on soil, normal flora, total plate count method was carried out. And the result are shown in this criteria was totally normal, and total count of normal microorganism in the soil was also checked. Next. These are the result of total plate count of soil during biodegradation at various time interval. Next. And last, I conclude that isolate JR7 was identified as Azotobacter corococcum on the basis of its cultural, morphological, and genetic analysis. And addition of this different concentration of silver nanoparticle into plastic film was significantly improve the shelf life of the packed food. Next. A total 12 types of agricultural waste sample were used for different sources, out of which 6 samples were potent cellulose source. And the highest yield I was found from the sugarcane bagasse, that is 0.98 gram per liter. Next. By comparing the both methods, that is a chemical method and that of microbial method, chemical method was more effective because microbial method, bifunctional molecule as a reducing agent and stabilizer was effectively changed. Next. The implementation of new active and biodegradable packaging technique with improved functionality and applied to increase the shelf life of food was also, I found that. And using this packaging strategy, we can stop the perishable food up to, we can store the perishable food up to 5 days and non-perishable food up to 15 days. Next. Various biological method was used for the synthesis of silver nanoparticles, and I used bacterial synthesis, and I produced the nanoparticles from Bacillus subtilis within 48 hours. Next. Thank you.
Thank you very much, ma'am. Thank you. It was an excellent presentation. Ma'am, I would now like to open us for the question and answer session. So in the audience, if anyone has any questions, kindly unmute yourself and please, you may ask the question. Do we have any questions in the audience?
Yes, sir. I have a very good morning and very Happy New Year to one and all. Madam, it was a wonderful session and you have given so much valuable information about your work. Ma'am, I want to know just one question thing, that basically when we are producing film, we are using PHB also. So how you compare that this cellulose based material which we are using for the formulation of film is better than PHB?
Yes, we can also use PHB, polyhydroxybutyrate, for the comparison. But from comparatively study shows that some review survey was show that from PHB is cellulose is far better than PHB, because after film packaging or food film development, the biodegradation of cellulose was very easy compared to PHB. So that we can comparatively study shows that cellulose is very useful and easy to biodegrade compared to PHB.
Okay, thank you so much, ma'am, for answering my question. Thank you.
Thank
Welcome.
you, ma'am. Thank you. Please, madam. Mahajan, ma'am.
Yeah, good morning to everyone, and I wish you very happy and prosperous new year. And I am so happy to hear from Madam about the work she has done. It's amazing and very novel work that is like in demand. So I had
Yes.
just a few points to tell you. In the slide where you mentioned acid treatment and alkali treatment, you showed one slide, I felt like
Yes.
there is some by mistakenly you have for acid you have written NaOH and
Oh, yes, yes,
some
yes.
other, so please
Yes.
make
Yes,
corrections
yes,
there
I
so that
corrected.
whenever you
Okay.
present, so next time when you present, that will not have a
Sure, sure.
and one place you said multiple sequence for J7.
Yes.
Yeah, that is, I think, your 16S RNA sequence that you have received, but by mistakenly you told like it is a multiple sequence analysis.
Okay.
Yeah, and otherwise presentation was wonderful.
Thank
Thank
you.
you
Thank you,
so much.
ma'am.
Thank you so much.
Thank you.
And thank you, ma'am, for those much needed feedback and comments. Thank you. Ma'am, I would request you to please write your email ID in the chat box.
Yes.
If you have any more questions, please unmute yourself and you may ask the question. Okay, your email ID is visible to everybody. So Chinmale, I think he is a student and he is requesting for PPT. So I advise you to kindly get in touch with the ma'am via her email
Yes.
ID. She
Sure.
will respond accordingly. Jalpa ma'am, I have a question myself.
Yes.
During your PhD journey, were there any low moments during your PhD work? And if there were any major issues you found, how did you overcome that?
Yes, there are very up and downs in my PhD work, but I say that Dr. Valentina madam is very supportive, and she was helped me to come out from this up and down by healing process and various steps of service.
Thank you, and thank you very much. Indeed, Valentina ma'am is very supportive faculty, I have heard.
Yes.
Thank you. So once again, Happy New Year to one and all. I would like to remind everybody that in this PhD webinar series of Microbiology Society of India, we are giving memento and certificate to every speaker and every student who have completed their PhD in past 5 years are welcome to present their work on this platform. So all the audience who are watching this, and we are also doing live streaming on YouTube, I request you to pass on a message to all the recent PhD students to please get in touch with us so we can register them for this webinar series and they can present their work to the world. Thank you. Thank you one and all. Thank you, Jalpa ma'am. And thank you
Welcome.
everybody. Thank you everybody for being here. We will meet next Sunday. And also we have one more lecture today 5 p.m. in the evening from Microbiology Society of India. So see you all. Thank you. Goodbye.
Goodbye.